Hello everyone, I'm Asma Yari. I'm the R&D Director at Nucleus Biologics, and today I will be introducing you to our QuickStart Media platform, which is an innovative approach to custom cell culture media that can significantly accelerate your development timeline and help you bring therapies to the clinic faster. So this is a really exciting time for the field of cell and gene therapy as we're seeing more creative approaches to address different angles of the therapies in development. With 37 approved cell and gene therapy products and over 4,000 therapies in development, the field of cell and gene therapy is getting higher attention from therapy developers, creating an opportunity for process improvement and reagent optimization. And when we think of it from the cells, the genetic modification to bioreactors and media, each piece of the puzzle influences the success of the final therapeutic product. And in particular, cell culture media, as it has proven its impact on cytotoxicity, cell potency, and ultimately therapeutic efficacy. Cell culture media isn't just a randomly selected component to include to your process, as it can either make it or break it. And it represents up to 25% of cost of goods, and it can directly influence your critical quality attributes. So that means early decision around media can have long-lasting implications on performance, regulatory outcomes, and the resilience of your supply chain. So it's essential to design for success from the very beginning. And a common pitfall in early development is the reliance on off-the-shelf media. While it seems simple, it often creates hidden complexity later. These products are usually proprietary, making it difficult to optimize or scale. And this lack of visibility leads to higher costs and delays when you're preparing for GMP manufacturing and regulatory filings. So you would wonder why cell culture media matters. Well, it's mostly because it accounts for about 25% of COGS when you don't own the formulation, and it constitutes your cell source of energy and microenvironment, which will have a direct impact on the cells you're growing. So having visibility on its composition is key to your success. Choosing the right media is a strategic decision. Off-the-shelf solution may help you move quickly at the start, but optimized media gives you full visibility, better performance tailored to your needs, and control over long-term costs. So media should be considered as critical as the cell type you're using, the genetic modification you're making, and the bioreactor strategy. So at first glance, off-the-shelf media might seem like the easiest path forward. It's available, it's been used before, and it gets you into the lab quickly. But that convenience comes at a cost, especially as you move towards clinical and commercial scale. So the core issue with off-the-shelf media is transparency. If you don't know what's in your media, you can't fully optimize your process. And when you hit performance challenges or run into regulatory questions, you're stuck working around a black box. So this lack of transparency leads to a number of downstream problems, such as the lack of optimization of yield and quality efficiently. You often pay for components you don't need, and you're likely locked into a sole source supplier, which is a major risk in GMP manufacturing. On the other hand, an optimized or custom media solution gives you control. You understand every component, you can tailor it to your cell type and process, and you're in much stronger position when it comes to cost, performance, and regulatory readiness. So the real question is, are you designing for speed today or for success tomorrow? QuickStart Media is our series of customizable, pre-optimized formulations developed specifically for key cell types like MSCs, HEK cells, and T cells, as well as cryopreservation. All our QuickStart media are xeno-free and serum-free. With QuickStart, you can rapidly prototype and optimize without sacrificing quality or transparency, enabling faster iteration cycles and smoother transitions to production. QuickStart Media helps you move from months of development to just weeks. With our ready-to-use media frameworks, you get a solid foundation that is customizable to your specific needs, speeding up iteration cycles and driving faster optimization. We currently offer a robust lineup of starting formulations. We have NB-MSC for mesenchymal stem cells, HEKima for adherent HEK cells, NB-ROC for T cells, and we also offer two cryopreservation formats. We have NB-KUL 10, which contains 10% DMSO, and NB-KUL GF, which is our DMSO-free option. Each is rigorously tested and designed to streamline development. And what's powerful about QuickStart is how easily you can iterate. QuickStart Media gives you the tools to easily modify formulations based on your process. So whether you're trying to address CQA gaps or remove certain components or tailor media to match your protocol, our platform simplifies the process. And you will benefit from complete formula transparency, robust supply chain support, and guidance from our scientific experts. So here's how it works. You choose a starting formulation from our QuickStart portfolio. Then we collaborate to modify it as needed. We start with an NDA and a clear project scope, and from there, we collaborate through 3 to 5 rounds of R&D. Once we land on the right formulation, we handle the tech transfer and help you scale up to GMP. It's fast, efficient, and designed to align with your therapeutic timeline. This gives you a head start, shortens development time, and delivers the custom performance your process demands. So what sets QuickStart Media apart is the combination of speed, flexibility, and transparency. You're not starting from scratch, and you're not locked into a black box formulation. Instead, you have a head start towards clinical readiness with the ability to control costs, improve CQAs, and reduce regulatory risks. So as previously mentioned, we have 5 ready to optimize formulations for cell expansion and cryopreservation, with 2 additional formulations soon to be added to the family: HEKima suspension to expand HEK cells in suspension, and NB-NK media for natural killer cells expansion. So for the next slides, I'll be sharing some of the data we generated for each one of the currently available formulations. NB-KUL DMSO-free or NB-KUL DF is part of the QuickStart family, and it's a DMSO-free, chemically defined, serum-free, and xeno-free cryopreservation medium that was tested on PBMCs, T cells, NK cells, MSCs, and HEK cells. So for customers seeking DMSO-free options, NB-KUL DF is an exceptional alternative. It matches or exceeds the performance of DMSO-containing media and clearly outperforms other DMSO-free competitors. NB-KUL DF effectiveness was tested on peripheral blood mononuclear cells, and this set of data shows equivalent performance of NB-KUL DF reflected by equivalent viability, cell count, expansion after thaw compared to DMSO-containing formulation. And more interestingly, we see a better performance from NB-KUL DF compared to DMSO-free counterparts. T cells were isolated from PBMCs and were cryopreserved in either NB-KUL DF, CS5, or CryoStor CSP. NB-KUL DF, as you can see on the graph, showed equivalent performance to CryoStor CS5 and better performance compared to CryoStor CSP in terms of cell viability and cell count after thaw on multiple donors. To further evaluate its performance, NB-KUL DF was used and tested on mesenchymal stem cells. And as the data shows here, NB-KUL DF showed a comparable performance to CryoStor CS5 containing 5% DMSO, but a better performance compared to CryoStor CSP in terms of cell count, viability, and expansion after thaw. And when tested on PBMC's isolated NK cells, NB-KUL DF performance showed slightly lower compared to DMSO-containing media in terms of cell count and viability on 3 donors after thaw. However, cell expansion showed equivalent. While compared to its DMSO-free counterpart, CryoStor CSP, it is still showing better performance. And this data is a good example to show that the media needs to be specifically developed for certain cell types to accommodate their specific needs, and that in this situation, media optimization is the answer to improve NB-KUL DF performance on NK cells. NB-KUL-10 is a high-performing DMSO-containing serum-free and xeno-free cryopreservation medium. So whether you're freezing MSCs, HEK cells, T cells, or NK cells, you will get a strong viability, recovery, and post-thaw proliferation, giving you flexibility and confidence in long-term storage. NB-KUL-10 performance was tested on mesenchymal stem cells for cell count, viability, and proliferation after thaw compared to CryoStor CS10. And as you can see, our data shows equivalent performance between the two tested Cryomedia. Interestingly, when tested on HEK adherent cells, NB-KUL-10 showed significantly higher performance compared to CS10 in terms of cell count and viability after thaw, and once live cells are plated for expansion, they showed comparable proliferation. NB-KUL-10 performance was also tested on T cells isolated from PBMCs. Across 3 donors, we were able to see equivalent readouts in terms of cell count, viability, and T cell expansion after thaw. NB-KUL-10 performance was also tested on NK cells compared to CS10, and we obtained equivalent performance for both Cryomedia in terms of cell count, viability, and also cell expansion. NB-ROC is a T-cell serum-free medium that was specifically developed to be used with T cells. And the data that we generated shows that it significantly boosts transduction efficiency while it maintains proliferation and preserves relative phenotype to support the development for CAR-T therapies. So our internal data supported by other labs' data showed that using NB-ROC for T-cell expansion leads to outstanding results compared to optimizer supplemented with either human serum or optimizer in enhancing over 4 times more T-cell transduction compared to off-the-shelf counterparts across 3 donors. In a head-to-head comparison, NB-ROC outperformed industry-standard media in terms of absolute GFP-positive cell count, which is a parameter that links between cell proliferation and transduction efficiency, which means that in your process you will end up having more efficacious cells. HEKima Adherent is a serum-free medium that was specifically developed to support HEK293 adherent cells expansion and boost protein expression levels compared to available competing media, resulting in a reduced viral vector and gene therapy manufacturing costs. HEKima Adherent has been tested across multiple HEK variants, and the data obtained is further confirming the need for customization. While it performed at equivalent levels to GMM plus 10% FBS for HEK293 and HEK293FT, it showed lower proliferation with HEK293T, which means having access to formulations developed for a specific cell type does not mean it will show the same performance across different variants of the cell line. HEKima performance in terms of cell expansion was tested as well compared to 2 competitors, sponsoring 293A and Pro 293, and the data shows superior proliferation when the cells are expanded in HEKima Adherent. To compare protein expression on cells grown in HEKima versus GMM plus 10% FBS, a lentivirus expressing GFP was used to transduce the cells. The data obtained shows a very significant increase in GFP expression when the cells are grown in HEKima Adherent compared to the control. So whether you're doing protein production or viral packaging, this media can deliver improved yield and consistency without the variability of serum. NB-MSC is a serum-free, xeno-free medium developed specifically for MSCs to support cell therapy and regenerative medicine research, as it offers a more consistent and reliable alternative to FBS supplemented media. So the data we generated shows that NB-MSC supports robust MSC proliferation compared to basal media supplemented with either 5% FBS or 10% FBS. And as presented on this graph, we see a significant improvement on proliferation kinetics and cell confluency when the cells are grown in NB-MSC, providing a defined scalable solution for stem cell culture and expansion. Evaluating the stemness of the cells comparing cells grown in NB-MSC versus DMAM plus 10% FBS shows that NB-MSC supports robust MSC proliferation while maintaining key pluripotency markers like CG90. So to wrap up, QuickStart Media is more than a product. It's a platform to accelerate your development. With high-performing, customizable formulations and full technical support from the formulation services team, you can reduce time, improve performance, and scale with confidence. Partnering with Nucleus Biologics, we can ensure consistent, transparent, and reliable media to fit your long-term strategy and help you get to the clinic faster. So beyond media itself, transparency and customization are core to our approach. You will always have full visibility into your media's components and concentration. Whether you need small volumes for testing or are scaling to GMP, we're with you every step of the way. Our leadership in quality, backed by XCPACT and ISO certification, means your media meets the highest standards from RUO to excipient GMP. What sets us apart is our end-to-end flexibility. We don't just offer off-the-shelf products, we work with you to fine-tune the formulation, validate performance, and manufacture at scale. Whether you're starting from scratch or need to refine an existing formula, we tailor the process to your needs while maintaining quality and speed. So thank you for your time today. If you're ready to take the next step toward optimized media, please feel free to reach out to us at sales@nucleusbiologics.com or visit nucleusbiologics.com. We're here to support you every step of the way as you bring your therapies to life.